Welcome back in another video tutorial from microbiology with Shagnik. So today we are going to start the Agrobacterium mediated gene transfer. Although we have also discussed about Agrobacterium mediated gene transfer in little bit in our earlier videos. So from that video I am going to develop a total lecture series regarding Agrobacterium tumefaciens, which will be consist of 6 videos, and it will cover entire thing that you need to know from Agrobacterium tumefaciens mediated gene transfer, okay, from 0 to hero. So let's start. So this is just the introductory video where we are just going to see what is actually the thing that is going on in case of Agrobacterium tumefaciens mediated gene transfer. So let's start. So in this method, suppose it is a tree and inside the soil there are two kinds of bacteria, namely Agrobacterium tumefaciens and Agrobacterium rhizogens, that typically employed in introducing any kind of foreign gene inside the plant cell. So Agrobacterium tumefaciens and Agrobacterium rhizogens are employed in introducing any kind of foreign gene inside the plant cell. Okay. Among them, the most important one that involved in any kind of gene transfer to plant is our Agrobacterium tumefaciens. Agrobacterium tumefaciens. This Agrobacterium tumefaciens is a gram-negative soil-borne pathogen. Okay, and this soil-borne gram-negative bacteria have the ability to cause crown gall disease in plants by synthesizing phytohormones that interferes with the normal growth of the plant development, and how we are going to see. So remember that Agrobacterium tumefaciens transfer a distinct portion of its DNA, or more specifically I should say a distinct portion of its plasmid's DNA. Okay, so this is the plasmid, TI plasmid, that is present in Agrobacterium tumefaciens, and this plasmid is generally involved in the gene transfer mechanism. So that distinct portion of DNA is known as tDNA, and that is why this kind of transfer is also known as tDNA transfer. So two DNA, or I should say gene, is involved in the entire tDNA transfer, or the involvement of two DNA is very important for the successful transfer of tDNA inside the plant cell. So what are they? First of all, the oncogenic tDNA itself, that is involved in the production of cancer, or I should say the tumor. Okay, and another one is the virulence region or vir genes. Okay, so they are involved in tDNA transfer. Now another thing that you need to know, that this left border and right border are directed repeat flanking sequence of 25 base pair. Okay, so this left border and right border play a important role in the tDNA transfer mechanism. Why? We are going to see in our upcoming videos, where I am going to show you that how the vir proteins produced by vir genes involved in react with the right border and left border and result in the release of a single stranded tDNA that will be guided by VD2, a protein inside the plant cell and inside the plant cell's nucleus. Okay, so what we have learned, that among the TI plasmid, virulence region is important for tDNA transfer, and tDNA itself is important, and among the tDNA, the left border and right border, which is a direct repeat of flanking sequences of 25 base pair, involved in tDNA transfer, or it determines the tDNA transfer. Now this tDNA will go, as you can see, this tDNA will go just like that and become integrated inside the plant cell, and by integration of the plant cell they start secreting three hormones. Okay, one is auxin, another one is cytokinin, and another one is opine. Among them, auxin and cytokinin are involved in interfering the normal growth of the plant cell. Okay, and opine is an unusual amino acid which is not utilized by plant cell. Okay, and so as a result, opine is believed to be utilized by Agrobacterium tumefaciens back. Okay, because as you can see, there a opine catabolism region is also present, and opine metabolism region is also present, right? So we can conclude that opine synthesized by tDNA in the plant is in turn going back to the Agrobacterium tumefaciens, and this opine is utilized by TI plasmid. Okay, so there are two kind of opines, nopaline and octapine, we are going to discuss, and depending on their types, different type of TI plasmid is present. Okay, now this, look at that vid regions. Okay, this virulence region also known as disarm strain. Why? We are going to discuss it when we will transfer our gene of interest with the help of co-integrative plasmid mechanism. We are going to discuss. Now this vid genes is present in, or is organized in different operons of TI plasmid, and we need to remember the name of that operons. So the name of that operons is vid A, vid B, vid C, vid D, vid E, vid F, vid G, vid H. In case of TI plasmid, along with TI plasmid, there are other operon present inside the chromosome, that is CHAV A, CHAV B, and CHAV F. CHAV A, CHAV B, and CHAV F. So this operon, in that operon our vid genes are organized, but we need to just remember about vid A, vid B, vid C, vid D, vid E, vid G, vid H. The operons present in the chromosomal region of vid gene we don't need to remember. Now there are the entire tDNA transfer mechanism by Agrobacterium tumefaciens to plant cell involves several steps. First is bacterial colonization of tDNA complex, then the tDNA transfer, later the integration of the tDNA into the plant cell nucleus. That is how the entire things going on. So this is the introductory video of Agrobacterium tumefaciens mediated gene transfer, that is commonly used to transfer any kind of gene of interest to plant. Here we generally used Agrobacterium tumefaciens. Okay, and Agrobacterium tumefaciens, mainly the TI plasmid present inside the Agrobacterium tumefaciens, play an important role in the entire transfer mechanism. Usually the TI plasmid involved in causing crown gall disease in plant by simply transferring the tDNA with the help of the vid genes that are present inside the TI plasmid, and after upon integration with the plant's whole or entire genome, tDNA starts synthesizing three genes, three product. One is auxin, another one is cytokinin, and another one is opine. Opine is go back to the Agrobacterium tumefaciens and utilized by them, and auxin and cytokinin are phytohormones, and that involved in interfering with the normal development of the plant cell. Okay, so that is how the entire process is going on. Now by looking the entire procedure, scientists thought that they can use that entire procedure as a gene transfer mechanism by just simply removing the tDNA, which is usually a not good for plant. Okay, so if we, with the help of recombinant DNA technology, can delete the tDNA from the Ti plasmid and insert our gene of interest, which is obviously good for plant, then this good gene will be inserted inside the plant nucleus with the help of virgins as a mechanism that they usually used in transferring the tDNA. So that is how the entire gene transfer mechanism is developed. Okay, and remember one thing, that tDNA transfer is determined by the left border and right border, which is directed sequence and flanking sequence of 25 base pair. So that's it for today.